So today we're going to start talking about genetics. So let's at least start off by getting a basic definition of what that means. So when we're talking about genetics, it's this entire field of biology that is interested in studying how traits get passed from one generation to the next. What we're talking about is inherited characteristics. Why do you look like your parents? Why do you look like your siblings? Why don't you look like everybody else? And it's really become a particularly popular branch of science, especially I'd say in the last couple of decades, as we've begun to understand what DNA is, how we replicate it, and how we actually use it via protein synthesis. So a lot of that stuff we've just talked about in this past unit, now we're going to kind of come out, take a look big picture, and look at what's going on with all of those processes and how you see them getting expressed at an entire organism level. So let's kind of start off with just a little bit of history. So you've got two particularly famous scientists, geneticists, Gregor Mendel, who is this gentleman right here. All right. He was the sort of father of genetics. He was working in the 1800s. He was a monk and is particularly famous for studying his pea plants. And he's really going to be the one who lays the foundation for us understanding how traits get passed on from one generation to the next, and why not every organism necessarily shows the exact same variation on a particular trait. So we'll walk through his pea plant experiment in just a moment. Another gentleman who has made some major contributions to the field of genetics is this guy down here, Thomas Hunt Morgan. And he really sort of took the field of genetics to a whole nother level in the 1930s. And most of his genetic studies were based on using these little itty bitty insects called fruit flies or Drosophila. And he is able to take the work of Mendel and really expand on it and show us that not every characteristic necessarily gets passed as a this or that sort of option, that there are some unique patterns to it. And using those fruit flies was actually a really great model for studying genetics, mostly because they're these tiny little organisms, they grow relatively quickly, you can get a new generation about every 2 weeks, and they have relatively simplistic chromosomal structure. So they've only got a couple of chromosomes, which is nice to work with in terms of figuring out where traits are actually located. If I were to look at a chromosome itself, is it on chromosome 1? Is it on chromosome 2? Where on that chromosome is it located? How are genes connected to each other? And so we'll talk a little bit today about crossing over and gene mapping, and he's really the one who managed to figure that out, and it's what ultimately got him a Nobel Peace Prize in 1933. So let's make sure we've got a couple of definitions on the books before we move any further today. So when we talk about inheritance or heredity, we're talking, at least from a biological standpoint, of this genetic transfer of characteristics from parent to offspring. So if we look at our cute little puppies here, if we were to mate these two dogs together, what does their offspring ultimately look like? What characteristics do they get from one parent as opposed to the other? So we can talk about things like hair color, eye color, skin color, but we can even go down to more specific levels and talk about, well, what sort of proteins got passed on that make you not only just look the way you do, which is typically how we teach this to you because it's something tangible that you can see, but also how you function. So do you have lactose intolerance? Are you missing that functional lactose gene? Other genetic diseases and the way that those particular mutations, something like breast cancer, might run in families because we're inheriting that DNA molecule as a whole, and then your body needs to express that set of information when you go through protein synthesis and start making those proteins from that DNA set of instructions. We also talk about this term trait, or you may come across it as simply just calling it a characteristic, but a trait in general is some sort of variation on a particular characteristic. So blue eyes versus brown eyes, do you have blonde hair versus brown hair, that sort of stuff. In our dogs, same idea, height, build, fur color, all of those are variations on particular characteristics. So I said we were going to do a little history today. So let's talk Mendel's pea plants, since this really is the first genetic study that sort of broke through and got us some insight into how traits get passed from generation to generation. So Mendel's considered the father of genetics because he runs these relatively simplistic, but at the same time, sophisticated experiments. So he's working with these pea plants. And all he's doing is growing tons and tons of plants in this greenhouse in his monastery. And he happens to notice, being a son of a farmer and things like that, that these pea plants happen to have very contrasting traits. Some of these plants will come up with round or wrinkled seeds. Some of them will come up with different colors, so yellow or green. Some of them will have different flower colors, different pod shapes. And so he starts exploring, well, what if I mate plants together, so I cross-pollinate them with different contrasting traits? What are their offspring, what do their progeny ultimately look like? So one of the things he recognizes is that he has to control the cross. He has to control which plants fertilize which. So with pea plants, if you take a look, all right, here is just a little background on flowers. So pea plant flowers happen to be what we call complex flowers, and all that means is that they have both female and male parts in the same flower. So if I look here, all right, here is this pistil, that's the female part of the plant, all right, and then these stamens, which are these guys, that's where the pollen comes from, are the males. So FYI, if you are allergic to plant pollen, you happen to be allergic to plant sperm. All right, so if he were to have left this plant go and just kind of do its thing, those stamens will ultimately produce pollen. That pollen will then end up fertilizing that pistil, and you will get the next generation of pea plants because they'll produce those peas, and those peas are the seeds to make the next generation. So what he ends up doing is something that in and of itself is relatively sophisticated. He recognized that he needed to not only control the crosses he was creating, but also needed to start off with what we now talk about as purebred plants. So what does that mean? If we're talking about a purebred plant, that means every time I make the next offspring, it will give me the exact same trait. There is no variation from it because that plant doesn't have an option to give any other characteristic. So if I'm looking at a plant, for example, that is a purebred purple flowered plant, that means that every time I mate two purebred purple plants together, I'm going to continue to get purple flowers in every offspring I ever produce. There will be no other option because those plants, those parents, don't have the instructions to give anything else but that purple color to their offspring. So once he produces these purebred plants, whether it was for flower color, pea shape, pea color, all of these different characteristics he ultimately ends up studying, he then crosses them on purpose. So he creates these purebreds and then comes in here and takes out either the male or the female part of the plant so that he can control which plants are mating with which. And all that means is that he was able to take, say, the stamens off of a purple flower, you use a little paintbrush, you dab it, pick up some pollen, kind of like a honeybee does when they kind of rub it in and move to the next plant, and then he'll take it to another plant and fertilize it. So he'll touch that pistil with that paintbrush that now has some pollen on there, and it will fertilize. So he's doing this in a very controlled environment. We talk about controlled experiments at the beginning of the year. You've talked about it as ninth graders. You've talked about it before that. And the whole purpose is that he is the one making sure that that's what is being transferred in terms of genetic material so that he knows where those genes are coming from. So he's trying to limit the amount of variability, the amount of different variables that are going on, so that only what he is testing for, his independent variable, is actually being shown in his data. So he performs a whole bunch of what we talk about as genetic crosses. So here is just the example with the flower colors. So he's got his two purebred parents. All right, what we talk about is your P generation. And those purebreds, every time I were to mate a purebred purple, I'm going to continue to get purple. Every time I purebred white, mate those guys together, I'm going to continue to get white flowers. And so he controls the experiment by purposefully making sure that he is mating a purebred purple with a purebred white. Puts those guys together, and what you get is your next generation, or what we talk about as your first filial generation, or F1 generation if we don't want to have to write that out all the time. And all that means is it's the first family generation. It's the next generation after those two purebred parents. And what he finds is that he gets all of these purple plants. Okay, that makes sense. But what he does is take it one step further. So let me give you a little background here. We're talking Mendel's point in history, and there's a lot of interest in how these genetic traits are getting passed. And most people, scientists or otherwise, recognized that if I mate two organisms together, I'm going to get some sort of combination of those parental traits in those offspring. We've been doing that with selective breeding in plants and animals, crops and livestock for thousands of years by this point. All right. So there's two big hypotheses being set on the table during Mendel's time. Do these traits get passed on as what they talked about as this blending hypothesis? So sort of like putting paint colors together. If I were to blend a purple and a white paint together, I'm still going to get purple back. It might be a little lighter, but it's still purple. So a lot of scientists were sort of on the side of that blending idea. I look at you as a human being and I look at your parents and you are some sort of combination of their two traits. You're a blend of their characteristics. The other side to that table was this particulate hypothesis, so particles. Were these traits being passed on as individual pieces? And when we looked at you, you just happened to be the combination of those pieces, those particulates, those particles. And had Mendel stopped at this F1 generation, it would have appeared to have supported that blending hypothesis. I put purple and white paint together, I get purple. What makes Mendel's work particularly useful is the fact that he took it one step further. So rather than stopping here, he took two of those F1 generations, all right, where we're going to realize they're hybrids or heterozygotes, and he produced one more generation. He produced this F2 or second filial generation. And here is where the support for that particular particulate hypothesis comes up. All right. And in fact, we know this is how this works now based on genetic studies, DNA studies, is that your parents give you that combination of characteristics. We go through meiosis, right? We make those sex cells. We separate out those traits, and then they come back together when they make the next generation. If this were truly just a blending hypothesis where you are a blend of your parents, then we'd never get this purple guy back. And in fact, if you were to look at any sort of species, we would all look identical. Now, you may say, well, a cardinal looks like a cardinal. Well, yeah, okay. But if you were to spend time studying those individuals, you'd start to see their differences too. But take a look at the human race. There's probably the easiest way to see all of those different variations that we have. If this were a blending hypothesis, the amount of generations that we've gone through as a human species other than maybe having two separate genders, we'd all look the same, and we don't look that way. So Mendel's able to very elegantly point that out, support that with these P plants. Okay? So I get that what we'll talk about as recessive trait, I get that guy back. All right? If this were the blending, it would never come back. Think of trying to pull out paint colors again. If I put purple and white paint together, good luck getting the white paint back. It's just not easily done if it's done even feasibly at all. So what ultimately comes out of Mendel's genetic studies? Well, he comes up with these 4 big hypotheses for inheritance. And we're going to spend most of this unit talking through those hypotheses and then what are the actual exceptions to his rules. Things that he didn't understand but geneticists that have come since him have figured out. So let's start with the first big hypothesis. Mendel's the first person to use this term called an allele. So when I look at characteristics, what are their alternative forms for the exact same trait? So if I'm looking at something like eye color, well, the different alleles would be your allele for blue, your allele for green, your allele for brown, so on and so forth, right? Those are the different variations for the same characteristic. If we're talking hair color, same idea. I've got an allele for being blonde, an allele for being brown-haired, an allele for being a redhead. Okay, they're all different forms for the same trait. And when I look at the actual genetics that are going on in your cells, we're looking at the fact that you've got those homologous chromosomes. Remember that? So I've got a chromosome 1 for mom and a chromosome 1 for dad. And at each position, or what we call a gene's locus or gene's loci, all right, I've got the same corresponding information on mom's chromosome that I also have on dad's. So here's a stretch of DNA, all right, sequence of A's, T's, C's, and G's on mom's chromosome that she now passed to her child. And let's just say that that allele happens to be for brown eyes, okay? So mom's giving the dominant and dad is giving the exact same sequence of A's, T's, C's, and G's for brown eyes. They are located at the exact same location on the exact same chromosome. Now I go to the next trait and let's assume that this is for hair color. Well, mom is passing on this particular trait. All right, well, we'll designate shorthand as a lowercase b. Hopefully you come across this as a recessive trait. And so her sequence of A's, T's, C's, and G's in that stretch of DNA at that particular location does happen to be different than what is on dad's homologous chromosome, the chromosome he gave you. All right, slightly different sequence of A's, T's, C's, and G's because now he's giving you the dominant. So let's assume brown hair is dominant, blonde hair is recessive. You now have to work with that combination of 2 proteins that are going to get produced from 2 different sequences of DNA, and that combination or that interplay between those proteins is ultimately the trait that you end up expressing. Hopefully that makes sense. So just a quick reminder, right? Homologous chromosomes are those matching pairs. So we have a total of 46 chromosomes, but really we should be talking about them as 23 pairs of chromosomes because you get 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, each of which came from mom and then came from dad. And so it's the interplay between the proteins that both of their genetic sets gave you that ultimately gives you the characteristics that you have. So what we're really talking about here is this idea of chromosomal theory of inheritance. So the genes that are located on your chromosomes, so the sequence of A's, T's, C's, and G's, and the behavior in which those chromosomes separate out during meiosis, right? So you should have one complete set, one chromosome 1, one chromosome 2, one chromosome 3, and one gamete, one sex cell, and the other set goes to the other sex cell. And then how those randomly come back together when we fertilize an egg plus a sperm to make the next generation is ultimately what accounts for all of the inheritance patterns that we see. So if I'm tracing where genes are located, those genes should be at the same location, or we'll now use the term a gene's locus, on those homologous chromosomes. So if I'm looking for the gene for hair color, it's going to be on the same chromosome from mom as it is from the chromosome you got from dad, and they should be roughly the same distance from one gene to the next, from the top of the chromosome to the bottom of the chromosome. Right? So let me kind of put this in perspective here. So here's back to Mendel's pea plants again for a second. So I've got two homologous chromosomes going on here. Here's chromosome 1, here's chromosome 2. Chromosome 1, we're just going to follow the gene for pea color, and it happens to be located here. If I look at, for pea shape, if I look at the gene for pea color, it happens to be on a completely different chromosome and at a different location. These are what is located currently in this mature plant. Now, this mature plant is going to eventually go to make sex cells, whether they be egg or sperm, to make the next generation. So when I go through meiosis, right, we're going to duplicate both of these chromosomes. We may get some crossing over that happens. We'll go through meiosis 1. We'll separate out those homologous chromosomes so that those diploid cells become haploid. And then from there, those sister chromatids, those duplicated copies, then split to make my 4 gametes. So I've got 4 different options for the different types of gametes I can create based on what my parent plant started with. So I can have 2 dominants. I can have a dominant for that shape, recessive for that color. I can also have the reverse. I can have the recessive for the shape, but dominant for the color, or I can have 2 recessives. And then it becomes a question of, well, which one of these ultimately gets fertilized to make that offspring for the next generation? All right, and that in and of itself is random. Another way to look at it, right? So here's 2 homologous chromosomes. This one came from dad, this one came from mom. Same location for pea color, on the same homologous chromosome. All right, so same locus. But mom and dad are giving slightly different variations on that trait. Why does this